Hello. My name is Alex, and I'm here to tell you about endoplasmic reticulum, rough and smooth, and sarcoplasmic reticulum. The endoplasmic reticulum, is an organelle of cells in eukaryotic organisms that forms an interconnected network of tubules, vesicles, and cisternae. Rough endoplasmic reticula are involved in the synthesis of proteins and is also a membrane factory for the cell. Smooth endoplasmic reticula are involved in the synthesis of lipids, including oils, phospholipids and steroids, metabolism of carbohydrates, regulation of calcium concentration and detoxification of drugs and poisons. Sarcoplasmic reticula solely regulate calcium levels.
The general structure of endoplasmic reticulum is a membranous network of cisternae, sac-like structures, held together by the cytoskeleton. The phospholipid membrane encloses a space, the cisternal space, or lumen, which is continuous with the prenuclear space but separate from the citosol. The functions of the endoplasmic reticulum vary greatly depending on its cell type, cell function, and cell needs. The can even modify to change over time in response to cell needs. The three most common varieties are called rough endoplasmic reticulum, smooth endoplasmic reticulum, and sarcoplasmic reticulum. The quantity of rear and cell in a cell can quickly interchange from one type to the other, depending on changing metabolic needs. Transformation can include embedment of new proteins in membrane as well as structural changes. Massive changes may also occur in protein content without noticeable structural changes. Rough endoplasmic reticulum. The surface of the rough endoplasmic reticulum, rear, is studded with protein manufacturing ribosomes, giving it a rough appearance, hence its name. The binding site of the ribosome on rear is a glycoprotein receptor called ribofurine. However, the ribosomes bound to the rear at any one time are not a stable part of this organelle structure, as ribosomes are constantly being bound and released from the membrane. A ribosome binds to the only when it begins to synthesize a protein destined for the excretory pathway. The membrane of the rear is continuous with the outer layer of the nuclear envelope. Although there is no continuous membrane between the rear and the Golgi apparatus, membrane-bound vesicles shuttle proteins between these two compartments. The rear works in concert with the Golgi complex to target new proteins to their proper destinations. A second method of transport out of the is areas called membrane contact sites, where the membranes of the and other organelles are held closely together, allowing the transfer of lipids and other small molecules. Smooth endoplasmic reticulum. The smooth endoplasmic reticulum, SER, has functions in several metabolic processes, including synthesis of lipids and steroids, metabolism of carbohydrates, regulation of calcium concentration, drug detoxification, attachment of receptors on cell membrane proteins, and steroid metabolism. It is connected to the nuclear envelope. Smooth endoplasmic reticulum is found in a variety of cell types, both animal and plant, and it serves different functions in each. The smoother also contains the enzyme glucose-6 phosphatase, which converts glucose-6 phosphate to glucose, a step in gluconeogenesis. The SER consists of tubules and vesicles that branch, forming a network. In some cells, there are dilated areas like the saxillary. The network of SER allows increased surface area for the action or storage of key enzymes and the products of these enzymes. Sarcoplasmic reticulum. The sarcoplasmic reticulum, senior, from the Greek sarx, flesh, is smoother or found in smooth and striated muscle. The only structural difference between this organelle and the is the medley of proteins they have, both bound to their membranes and drifting within the confines of the lumens. This fundamental difference is indicative of their functions. The synthesizes molecules, while the senior stores and pumps calcium ions. The senior contains large stores of calcium, which it sequesters and then releases when the muscle cell is stimulated. The SR's release of calcium upon electrical stimulation of the cell plays a major role in excitation-contraction coupling.